Hi, I'm Kimberly Gutierrez, Senior Field Application Scientist at Stilla Technologies, Inc. I would first like to thank the organizers of this conference for giving us the opportunity to present, and I'd also like to thank you for taking the time to watch this presentation on the use of crystal digital PCR to detect SARS-CoV-2 using Stilla's NICA system. For those of you who may not be familiar with digital PCR, we will start by reviewing its principles, followed by a description of the NICA workflow and the use of this technology to detect SARS-CoV-2. Digital PCR is the third generation of the well-known polymerase chain reaction, or PCR, a widely adopted technique in the molecular biologist toolkit. PCR allows for the detection of targeted nucleic acid sequences by specifically amplifying them in solution. By following the evolution of an increasing fluorescent signal as PCR occurs, the so-called quantitative version of PCR, qPCR, allows for the quantification of the targeted sequences as well. Although easy to use, qPCR is a relative quantification method, as it requires the establishment of a calibration curve with standard samples of known concentration. These standards may not amplify with the same efficiency as the sample of interest, which may bias the quantification. By partitioning the PCR reaction mix, digital PCR enables an absolute measurement of targets concentrations, overcoming the limitations of qPCR and conferring an increased sensitivity. We will see how in more detail. In digital PCR, the workflow starts with the same reaction mix as for qPCR, containing the reagents necessary for the amplification reaction. The key step in digital PCR is the partitioning of the reaction mix into a large number of small compartments of the same size. In the NICA system, partitioning is generated by a water and oil emulsion of the mix. As the emulsion is generated, the target sequences are randomly distributed in the droplets, such that some droplets contain one or at least one sequence of interest, and some don't contain any sequence at all. After partitioning, each droplet acts as an independent reactor, where PCR can occur if the target is initially present. A successful PCR will lead to the generation of a fluorescent signal at the end of the reaction within the droplets that initially contained the targets of interest. The fluorescence intensity of each droplet is then plotted via a dot plot, with two clusters separated by a threshold automatically set by the analysis software. Above the threshold are the droplets considered as positive. Under the threshold are the droplets considered as negative. The analysis software then uses the proportion of positive droplets and Poisson statistics to estimate the concentration of the target of interest, together with the uncertainty on the measurement. The latter lies in the fact that a positive droplet may contain more than one target sequence initially, and is dependent on both the concentration of the target in the PCR mix and the number of droplets generated. The patented partitioning technology by contactless fluid injection in the NICA system leads to a self-assembled array of droplets called a droplet crystal. In a droplet crystal, the size of the droplets is fixed and controlled via the microfluidic design. Ensuring a monodispersed population of droplet size is important in order to guarantee a random distribution of the target sequences within the droplet crystals. The other specificity of the NICA system is that it has 3 fluorescent channels, allowing for the quantification of 3 different targets in a unique sample. In this scheme, for example, 2 variant sequences of genomic DNA can be quantified together with the wild-type sequence. In such an experiment, the lowest abundant target, in red here, is artificially enriched by partitioning, making it less prone to competition, as would be the case in a bulk solution in qPCR. That is why digital PCR is more sensitive than qPCR to detect rare events. Making digital PCR a lab commodity is one of our missions at Stilla, and the NICA system is an easy-to-use and integrated solution. The entire workflow takes no more than 2.5 hours and requires only a few manipulations, as we will see in more detail. First, let's focus on the heart of the innovation, the sapphire chip, which integrates the droplet crystal technology. It is already pre-filled with oil, Thus, the only manual step is to load the PCR mix in the chip. With an input volume of 25 microliters of PCR mix, you end up with droplet crystals between 25,000 to 30,000 droplets of 0.5 nanoliters each. Given these specifications, the dynamic range of the NICA system spans 5 logs, from an LOD at 95% confidence level of 0.2 copies per microliter up to 20,000 copies per microliter. It is possible to load up to 4 samples per chip. To load the chips with PCR mix, you simply have to open the inlet port, pipette 25 microliters of mix on top of the oil already inside, and seal the inlet port with a cap. In terms of compatible chemistries, you can run digital PCR experiments starting from DNA or one-step RT digital PCR experiments starting from RNA. And the system is compatible with TaqMan probes for fluorescence detection. Once loaded and sealed, up to 3 sapphire chips, so up to 12 samples, can be placed in the first instrument, the NICA geode, which will perform both the partitioning and the PCR cycling by generating the emulsion and then heating and cooling the droplet crystals following the PCR program defined by the user. At the end of the PCR reaction, you simply need to switch the chips from the geode to the second instrument, the NICA Prism 3, which will read the endpoint fluorescence signal of each crystal in the 3 channels. Thanks to the droplets forming a crystal, one picture is enough to catch the signal of every droplet, allowing a fast scanning time of about 50 seconds per sample. When multiplexing with TaqMan probes, custom probe combinations can be chosen as long as they match the wavelengths displayed here. The main fluorophores used in molecular biology are compatible with the system. After scanning, data visualization and analysis is performed by the Crystal Miner software. It has been completely developed in-house and allows a guided and intuitive analysis of the data for an in-depth understanding of the results, as well as a robust and reliable quality control. That includes, amongst other features, the Explore Crystal feature, where the dot plot data is presented alongside the associated droplet crystal image, and allows you to visualize every single dot and its corresponding droplet in the crystal. Crystal Miner is available with no limitation on users or licensing fees, and can be downloaded together with demo data via Stilla Technologies website. To sum up, the NICA system workflow allows you to achieve results in 2.5 hours with minimum hands-on time. The integration of the workflow within the Sapphire chip makes it robust against contamination and very easy to use. The NICA system is, of course, compatible with many sample types to address a wide range of applications. Advantages of this easy-to-perform digital PCR workflow will benefit your analyses for absolute quantification of DNA or RNA, copy number variation assessment, rare event detection, or gene expression analysis. One of the applications for digital PCR is in the field of infectious disease. So I'll now speak about the use of the NICA system in the detection of SARS-CoV-2. We are currently living through the first pandemic in over 100 years. The COVID-19 pandemic caused by the SARS-CoV-2 virus has greatly altered daily lives worldwide. As of April 28, 2020, there have been over 3 million confirmed cases of COVID-19 worldwide, of which more than 1 million are in the US alone. The US has conducted more than 5.6 million tests for COVID-19, but this accounts for less than 2% of the total US population. These statistics highlight the need for more testing of the virus. In order to combat the spread and a potential second wave of infection, there is also a need for more research to understand the viral pathogenicity, but also to understand the clinical outcomes of infection. Contributing to the battle against COVID-19, Stilla has partnered with Apex Bio to offer a digital PCR kit to detect SARS-CoV-2. Our partners at Apex Bio have developed a novel 3-color kit to detect COVID-19. The kit contains 2 viral genes, ORF1ab and nucleoprotein N, which are detected in the FAM and HEX channels respectively. A human housekeeping gene used as a control is detected in the red channel as Cy5. The kit contains all primers and probes, the RT digital PCR master mix, as well as COVID-19 positive and negative controls used to validate each run. And currently, this kit is for research purposes only. Data generated by Stilla's R&D exhibits the sensitivity and specificity of the kit to detect COVID-19 sequences. In this experiment, plasmids were used as positive controls containing the viral genes ORF1ab and nucleoprotein N. The positive control was serially diluted and tested in triplicate. 8 microliters of the positive control was assessed in a 25 microliter reaction in the presence of 1 nanogram of human RNA. The kit reliably detects viral sequences down to 0.6 copies per microliter of ORF1ab and 0.9 copies per microliter of nucleoprotein N. Further dilutions, not shown here, detected the viral genes down to 0.25 copies per microliter, or the equivalent of 2 copies per 25 microliter reaction. Importantly, no false positives were observed in 15 negative controls containing 1 nanogram of human RNA. Stilla recently collaborated with a group at the Institute and University Hospital of Mediterranean Infection in Marseille, France. Here, patient samples originally tested by qPCR were revalidated by crystal digital PCR in order to investigate whether the increased sensitivity enabled by digital PCR could be used to further verify the presence or absence of viral sequences in the samples with high CT values obtained by qPCR. The negative and positive groups, as determined by qPCR, were also revalidated. In the negative or undetermined group, crystal digital PCR identified and recalled 1 sample out of 15 as testing positive for COVID-19. The results from the positive group were used to try and determine a correlation between crystal digital PCR and qPCR results. However, a full correlation could not be reached as qPCR is not standardized and the patient samples were from different series. The doubtful population, or any sample with a CT value greater than 34, were retested by crystal digital PCR and given a designation of positive or negative for viral sequences. 7 of the 18 samples tested were designated as negative for COVID-19, while the presence of viral sequences was confirmed in 11 of the 18 samples tested by crystal digital PCR. This data demonstrates the high sensitivity of the crystal digital PCR COVID-19 detection kit and highlights its use as a powerful solution to investigate difficult to interpret high CT value qPCR data. To briefly compare digital PCR to qPCR, both processes share the same sample preparation methods and reagents, have similar sample volumes, and both methods are capable of multiplexing. When it comes to analysis, digital PCR does not require a standard curve or calibration of the results. Therefore, digital PCR allows for the absolute quantification of results with lower variability. And finally, digital PCR demonstrates higher sensitivity and reproducibility as compared to qPCR, rendering it a more effective technique to detect rare or low-level events. If you'd like to know more about specific applications, I invite you to visit our application and technical notes available on our website. Here, you can find an application note about the COVID-19 detection kit. And if you'd like to learn more about crystal digital PCR principles and applications, I encourage you to visit our online learning center at www.genepy.com. A special thank you to our collaborators for providing the SARS-CoV-2 detection data. And thank you for your time. I'm happy to answer any of your questions, or feel free to contact me via email. Enjoy the rest of the conference and stay safe.